Hello, and welcome to this RPTH research recap, where we dive a little deeper into the science being published at RPTH. My name is Robert Campbell, and I'm a member of the RPTH editorial board. And I'm also an assistant professor at the University of Utah in the Department of Internal Medicine. I've been at the University of Utah now for 13 years, where my research focuses on how inflammation alters megakaryocyte and platelet function. However, during my graduate work at the University of North Carolina at Chapel Hill under the direction of Alyssa Wohlberg, my research interests were on fibrin and how fibrin clots form. During this time, I began to admire the work of the senior author who is presenting his work here today. Therefore, I'm pleased to introduce to you Dr. John Weisel. John Weisel received his bachelor's from Swarthmore College and his PhD from Brandeis University. Research in the Weisel lab covers a variety of aspects of blood clotting, but the primary focus has been on molecular and cellular mechanisms of fibrin polymerization, fibrinolysis, and platelet aggregation. His laboratory studies the structure and mechanical properties, including rupture of human and mouse blood clots and thrombi from patients. They are particularly known for their use of various biophysical and structural approaches, including many novel imaging techniques that they have developed and they have also developed an optical trapping technique to study protein-protein binding and unbinding at the single molecule level, including the kinetics and thermodynamics. They investigate the molecular and cellular mechanisms of blood clot contraction and its role in hemostasis and thrombosis. In all of this research, there's an emphasis on the clinical implications for bleeding, thrombosis, and embolization. Dr. Weisel recently published a paper in RPTH titled Blood Clot Contraction, Mechanisms, Pathophysiology, and Disease, a state-of-the-art review of his research, which was presented last summer at the ISTH Annual Congress in London, which focused on how blood clots are formed and their pathological roles in disease. For those of you joining us live on Facebook, Twitter, and YouTube, please leave your comments below, which will be answered during the discussion. And Dr. Weisel, we look forward to hearing your presentation today.
Thank you very much, Robbie. And I'd like to emphasize here, this was a review article that was presented by my colleague, Rustem Litvinov, and we wrote the review article together. Also, I'd like to emphasize that this is an understudied area in thrombosis and hemostasis. So a lot of these conclusions I'm going to be giving today are somewhat tentative, and I encourage anyone who's interested to pursue research in this area because there's a lot yet that needs to be done. So this is on clot contraction, sometimes called retraction, which is the volume shrinkage of a clot that's driven by platelets pulling on fibrin. On the left here is whole blood in a test tube, and after it clots and contracts, you see the image on the right. And this process takes place over a period of about a half an hour in the test tube. Clot contraction is said to be significant in hemostasis to form a better seal, in thrombosis to restore blood flow past otherwise obstructive thrombi, and for wound healing. The molecular mechanism of contraction in platelets or in many other cells is shown here. Contraction is initiated by signaling processes when platelets are activated, and force is generated by non-muscle myosin 2A pulling on actin filaments inside the platelets, transmitting the force to fibrin outside the platelets via the integrin alpha 2B beta 3. This is a confocal image showing the cellular mechanism of clot contraction. And here you see platelet filopodia pulling on fibrin fibers, causing deformation. You can see this process a little bit more easily in these images where the two channels are separated. The platelet here in green is extending filopodia, which then grab a hold of a fibrin fiber. You see a kink as a result of the filopodia pulling, and then you see the fibrin agglomerating around that individual platelet shown there. So this is like what this fellow here who's pulling on a rope hand over hand. These filopodia are coming out, pulling on the fibrin fiber and accumulating fibrin around the platelets in the clot. If you look by scanning electron microscopy, at the outside of that contracted clot, you saw a few slides back, you see a very dense meshwork of fibrin and platelets. And if you look on the inside, you see a very tightly packed tessellated array of polyhedral red blood cells. Since they're polyhedral erythrocytes, we call them polyhedrocytes. And if you're looking by confocal microscopy at a labeled red, this is a single polyhedrocyte in a contracted clot, you can see this polyhedral structure that is present inside the contracted clot when the red blood cells are compressed by fibrin, by the platelets pulling on fibrin. If you take unclotted whole blood and centrifuge it, you see similar sorts of structures just from the forces of centrifugation. So there's nothing special about the clotting. It's just the forces generated by the platelets pulling on fibrin that compress the red blood cells, and their shape changes from biconcave to polyhedral. So we wanted to develop an assay that to quantify clot contraction. This is a simple assay using whole blood, and we measure the extent of contraction, but also the rates of contraction, and we can look at the effects of all the components, platelets, fibrinogen, red blood cells, and other blood plasma components. And this can be used for patient samples for clinical studies or for mouse blood. So this is the basic instrument that we use, and this is a light source on a cuvette that contains blood or platelet-rich plasma, and a CCD camera to capture the contraction of the clot as a function of time. And as a result, we see this sort of a curve where we can look at the endpoint, the extent of contraction at the beginning, the lag time before contraction begins, and the area under the curve, which is the work done by platelets. If we take the first derivative, then we can measure the rates, and we can see that there are three phases, and we can measure the rates of contraction in those three phases, and the length of each of those rates, of those phases. If we use this instrument to look at how clot contraction affects fibrinolysis or dissolution, enzymatic dissolution of a clot, we can see here with internal fibrinolysis, where TPA is present from the beginning, and this is a model system for physiological fibrinolysis, we see that the rate of contraction is enhanced, is increased by contraction. In contrast, for external fibrinolysis, where the TPA is present outside of the contracted clot, which is a model for clinical thrombolysis, we see that the rate of contraction is lower. So the clot contraction decreases the rate of external fibrinolysis or thrombolysis. Clot contraction has been studied in a number of thrombotic conditions shown here, and also in a number of bleeding conditions. I'm just going to show a few examples to illustrate how clot contraction affects these conditions. This is a model using a mouse saphenous vein, injury of the mouse saphenous vein for hemostasis, and then we can measure the bleeding time, but also look by scanning electron microscopy here at the resulting clot. And what we see is that there are areas where there are polyhedrocytes that are enclosed by dense meshwork of fibrin and platelet. So it's very similar to what we see in vitro with clot contraction. And if we look at clots from hemophilia mice, these mice have fewer polyhedrocytes, and so the clot is more permeable, and there's increased bleeding in the hemophilia mice. So as a tentative conclusion here, for hemostasis, we can say that clot contraction is necessary to form a tight seal, and with impaired clot contraction, we have increased bleeding. So if we look at an example for thrombosis, this is a study with ischemic stroke patients, and we see here that the extent of clot contraction is considerably less in these stroke patients than in healthy subjects, and the rates of contraction are also affected. If we look at the properties of the platelets from these stroke patients, we see that in resting conditions, there are a higher percentage of partially activated platelets. And if we activate the platelets with any kind of stimulus, then there are a lower percentage of activated platelets. So if we look at the fold activation for stroke patients, we see considerably lower extent of activation, which means that the platelets in these stroke patients have been partially activated. They become exhausted and refractory to further activation. So this is a schematic diagram that shows some of the tentative conclusions for the result of impaired contraction in thrombosis. So if there are thrombotic conditions, so you have local prothrombotic conditions and hyperfibrinogenemia, then that leads to hypercoagulability, systemic thrombonemia, which leads to continuous platelet activation. Platelets become exhausted and refractory and dysfunctional, which then leads to impaired contraction. And that it results in increased obstructiveness of thrombi. There is reduced internal fibrinolysis. So it's an antifibrinolytic mechanism. And there is a predisposition to embolization in these with impaired contraction. So this is a tentative conclusion for thrombosis, that clot contraction appears to be necessary to reduce obstructiveness of thrombi. And with impaired contraction, we have increased obstruction, which is detrimental in thrombotic patients. If we look at thrombi taken from thrombotic patients, these are from coronary artery thrombi taken from myocardial infarction patients, what we see are very tightly packed polyhydrocytes here. And these polyhydrocytes are present mainly in the upstream part of the coronary artery thrombi. So at the point of initiation, there are mainly platelets. And then as you go from initiation farther upstream, you see increased number of red blood cells, mostly in the form of polyhydrocytes. If we look, these are thrombi taken from ischemic stroke patients. You can see at the top left at low magnification, and then at the bottom left, increase in magnification and higher magnification on the right. And you can correlate the structure of these stroke thrombi with the clinical course and outcomes of disease. So as our conclusion here, this is summarizing in hemostasis with normal contraction. You have a low low permeability, whereas if you have defective contraction, there is leakage, more permeable clot and leakage of fluid through the less contracted clot. Whereas in thrombosis, you have contraction which reduces the obstructiveness of thrombi and it improves the blood flow. So this was a review article, so we cite the work from many different groups, but I want here to thank some of the people from my group and some of my collaborators who contributed to the work from my group that was shown here in this presentation. And I'll thank you for your attention, and I'm ready to answer any questions you might have.
Great. Thank you so much, Dr. Weisel, for your excellent presentation. I think one of the first questions I wanted to maybe address, and you addressed this throughout your summary, is sort of, maybe going a little bit more detail. What is the difference in clot contraction in these prothrombotic patients compared to controls?
We found in a lot of different conditions, we see a lower extent of contraction and slower rates of contraction. And also in all of the patients we've studied, in different patient groups, we see the contraction is impaired in thrombotic patients. And that appears to be a result of a whole variety of different things that happen. With inflammation, there is hyperfibrinogenemia, so higher fibrinogen concentration, which leads to impaired contraction, but also primarily changes in the platelets, where the platelets are partially activated and they become exhausted, metabolically exhausted when we look at ATP in the platelets, for example, and they're refractory, so that those platelets are impaired, and that's what leads to the impaired contraction in the thrombotic patients.
So, and when you were looking at the patients that have, bleeding disorders versus these prothrombotic patients, it appears that the clot contraction is defective similarly in both, but it's sort of where the location of the clot is. And maybe can you talk about that a little bit more? Because it seems like some of the mechanisms are pretty similar, but it's maybe the location of the clot compared to the quality of the clot.
Right. So this was a big surprise to us when we began these studies, and that appears to be the conclusion. But I want to emphasize also that this is still tentative. There's a lot still that needs to be done, and especially in hemostasis, there's much fewer studies of the effect of clot contraction in hemostasis. So, but our tentative conclusion is just as you said, that there is impaired contraction in hemophilia, for example, because of lower generation of thrombin, and there's impaired contraction in thrombotic or prothrombotic conditions. So that's our tentative conclusion at the moment. And it depends, as you said, it depends on the location of the clot or thrombus.
So, and you sort of touched on this on the answer to my first question. So what's the difference in platelets in these prothrombotic conditions patients compared to the controls?
Yeah, so this was, again, a big surprise to us when we started looking. If we look at various, by flow cytometry, various markers on the surface of platelets, what we see is that there are a higher percentage of activated platelets when you just take blood from the patient, that are platelets that are partially activated, and then if you activate the platelets, they're less activatable, so that they appear to be exhausted and partially refractory, so that the platelets are impaired in these prothrombotic patients.
All right, and maybe my last question for you is, I think in our field, a lot of people focus on the platelets, a lot of people focus on the fibrin and the mechanism of the fibrin, but how much of a role do you think the red blood cells play, and in terms of bleeding diatheses or thrombotic disorders, in terms of their ability to form? Are we missing things in terms of the biology of the red cells compared to like the things that we always focus on in platelets and fibrin and thrombin?
That's a very good question, and actually I mentioned that I also wrote a review article with Rustem Litvinov, my colleague, on the forgotten or unknown roles of red blood cells in hemostasis and thrombosis. So this is another understudied area. We know a lot for a long time about their role in terms of margination with flow, but red blood cells play many other roles. That they generate thrombin on their surface with phosphatidylserine exposure. But we also think that in certain types of clots and thrombi, they also play a role that you saw here in the structure of the clots, and particularly in venous thrombosis, where there are a lot of red blood cells. I think the contraction makes a much better seal, very highly impermeable seal, because of this shape change from biconcave to polyhedral, and I think this plays a role in hemostasis, and even in thrombosis, because those contracted clot is much less permeable, and in thrombosis, for example, I think this is why the thrombolytic treatments that have been used for many years are less effective than people thought they would be. And I mentioned venous thrombosis, but even in arterial thrombosis, we've looked at a lot of thrombi taken from patients both with myocardial infarction, stroke, and other conditions, and even arterial thrombi have a lot more red blood cells than we expected, and they also are mostly, very few are biconcave, they're mostly polyhedral. So we think that clot the red blood cells in clot contraction plays a role in both arterial and venous thrombosis, but probably more so in venous thrombosis.
Oh, great. So we actually have a question from Twitter. So within thrombotic patients, have differences in clot contraction been observed between patients? Has this been related to adverse or long-term outcomes? I think this is from Robert Lee at UNC Chapel Hill.
The long-term effects. And
so the question is, have there differences in clot contraction been observed between patients, and has this related to adverse long-term outcomes?
So I give one example here. We don't know so much long-term, but a study we did of venous thromboembolism, for example, we saw decreased clot contraction in these VTE patients. But in the VTE patients who also had pulmonary embolism, there was even lower extent of clot contraction and rates of contraction than in the patients who had isolated venous thrombosis in the lower limbs. So this is a difference between these two groups of patients, and so we can then probably relate the extent of contraction to embolization, that lower extent of clot contraction leads to a less stable clot or thrombus, and it's more likely to embolize. So we haven't done much, and there's not much in the literature on long-term outcomes, but this is one difference between different groups of patients that we have observed.
Oh, no, thanks, and thanks for your question. So I would like to just thank you, Dr. Weisel, for presenting your work here today and giving us some more insights into mechanisms of fibrin clot formation and contraction and hemostasis and thrombosis. And we hope viewers will read the full details of your paper on our site, rpth.isth.org. And we hope that we'll see some of your work in the future at RPTH. Thank you, Dr. Weisel.
Thank you. I enjoyed